Now, let's take a look deeper on the different components of nucleotides. One is the sugar or the ribose sugar. Okay, so in this structure, we have the pentose sugar. So why is it called as the pentose sugar? Because it has 5 carbons. Okay, so we have 1, 2, 3, 4, 5 carbons. So this is actually the general formula of a ribose sugar. Okay, so the 5 carbon sugar composed of these carbons. And remember each carbon here, the carbon number 2, 3, and 5 is actually the attachments of other elements or other compounds or the R groups later on. So in this carbon here, in the second carbon of the pentose sugar, we put it here an X that signifies if X is hydrogen, therefore the sugar is DNA, okay, or the deoxyribonucleic acid. If X here is OH, that means that is the sugar of ribonucleic acid. Okay, so for example, we have here, if it's H, that is 2' deoxyribose. If it's OH here, it's 2' ribose sugar. Another component of a nucleotide is the nitrogenous base. The nitrogenous base derivatives of either the pyrimidine or the purine. So we have two types of bases, as what I've said. It's purine and pyrimidine. When we say purine, it have the double ring structure, and the type of your purine is your adenine and your guanine, while we are talking with your pyrimidines have a single ring structure, and these includes the cytosine and thymine. Again, in this presentation, we have the general formula here. We note that if oxygen, it is always double bond. So meaning if that will be oxygen is attached on that X here, the X here, that is double bond. So for example, we have this pyrimidine or the single ring structure, has a single ring structure. So this is the general formula. We put X here and we put Y here. If the X in the structure here is NH2, that will be the cytosine. If the X is oxygen, the pyrimidine is uracil. If the X is oxygen and the Y is CH3, that means it is thymine. So let's try to look into this. We have here the cytosine. As what I've said, if the X here is NH2, then it will be cytosine. And the name for cytosine is 2-oxo-4-aminopyrimidine. Next, we have the uracil. If the X is oxygen and it is always double bond, it will be your uracil. And the name is 2,4-dioxopyrimidine. Lastly, if the X is oxygen and the Y is CH3, therefore that base is thymine, or it is known as your 2,4-dioxo-5-methylpyrimidine or 5-methyl-uracil. Next, we have the purine. Purine has a general formula of this one. Again, purine has two ring structure. And we have here the X and the Y structures. attached to your ring. If the X is NH2, therefore it is called as adenine. If X is oxygen and the Y is NH2, then it is guanine. Next component of nucleotide is your phosphate group. Your phosphate group imparts negative charge to the DNA. Okay, as what I've mentioned last time, if two phosphates, then therefore it's diphosphate. If one, it's monophosphate. If three, it's triphosphate. Again, your connection on the base here, the base and the sugar or five ring sugar or five carbon sugar, I mean, it is called as N-glycosidic bond. This is actually the phosphate bonds. So phosphodiester bond in the connection of our phosphate group and our sugar. In summary, these are the structures of your pyrimidines and your purines. So in the DNA, each base is chemically linked to one molecule of sugar deoxyribose, forming a compound called the nucleoside. When the phosphate now is attached to the sugar, the nucleoside will become nucleotide. Again, I will reiterate. In the DNA, each base is chemically linked to one molecule of the sugar, forming a compound called the nucleoside. Okay? Once phosphate is attached to the sugar, then the nucleoside will become nucleotide. Observe in this structure of nitrogenous bases, in each base, the nitrogen atom is linked to the deoxyribose sugar, which is indicated in this presentation or in this slide. Atoms with darker backgrounds participate in hydrogen bonding between the DNA base pairs. So here, the deoxyribose sugar is attached to the nitrogen, another one attached to this nitrogen. In the pyrimidines, the deoxyribose sugar is attached to the nitrogen. Also, in the cytosine, the deoxyribose is attached to this nitrogen. In this slide, the base is in the first carbon, which is conventional way the atom is sugar are given prime numbers to distinguish them from atoms in the base. In the nucleic acids, the nucleotides are joined to form a polynucleotide chain, and in which the phosphate attached to the 5' carbon sugar is linked to the hydroxyl group attached to the 3' carbon of the next sugar in line. So here, again, this is in the 5th carbon, naka-attach ang imo nga phosphate, and this 2 carbon or the 3' carbon will attach to another nucleotide later on. Notice also here that the chemical bonds by which the sugar components of adjacent nucleotides are linked through a phosphate groups are called the phosphodiester bonds. And the 5' end, which terminates in a phosphate here, and the 3' end terminates in the hydroxyl group. That is why it is called as 5' end terminates with the phosphate group, while the 3' end terminates with a hydroxyl group or the OH group. These areas are exposed where the next another nucleotide will be able to attach. on these areas. And that ends the presentation. Thank you very much for your kind attention.